So gibberellins are growth promoters that are involved in the stem elongation of pea plants. Now, this was all discovered by Gregor Mendel, and he found that the length gene, so that we write as capital LE and gene, codes for the production of an enzyme called GA3B hydroxylase. Now, this enzyme catalyzes the final reaction in a biochemical process to produce. Now, gibberellin then promotes the stem elongation of the pea plant, which results in a tall plant. Mutations on the LE gene result in a recessive allele. So it will be a lowercase LE, and it will be again gene, so the recessive allele. The recessive allele cannot produce a functioning GA3B hydroxylase enzyme, and so the gibberellin is not produced, and so stem elongation can't occur, and this results in a dwarf pea plant. Now, again, it's important to understand the LE, so capital LE is dominant and the lowercase le is recessive. So what this means is that if we have heterozygous and heterozygous individuals, so capital LE and then lowercase le is its genotype, the plant, the pea plant, will be a tall plant because the LE gene will mask the effect of the recessive allele. In a homozygous dominant, we have both dominant alleles, so we will have stem elongation. And then in homozygous recessive, both alleles will be the recessive allele, the mutated allele of the length gene, and this will result in no production of a functioning GA3B hydroxylase enzyme, so no gibberellin will be produced, and so stem elongation will not occur, and the pea plant will be a dwarf plant. Now let's take a look at the difference between repressible and inducible enzymes. So repressible enzymes are enzymes that are being constantly produced. Their gene is constantly switched on. And the way to turn a repressible enzyme off will be when a molecule comes and prevents the transcription of the gene. On the other hand, inducible enzymes are enzymes that are normally switched off, but in the presence of a specific molecule, their genes will be turned on and the production of these inducible enzymes will begin. Structural genes are genes that code for polypeptides, whereas regulatory genes code for proteins that switch on or switch off the structural genes. So, in the bacteria called E. coli, the main respiratory substrate is glucose. In this situation where the E. coli is grown in a medium of lactose, it produces 3 enzymes in order to survive, and these 3 enzymes are in the lactose operon or the lac operon. The first enzyme is beta-lactosidase, which is coded for by the gene called LAC-Z. Now, beta-lactosidase hydrolyzes the breakdown of lactose into glucose and galactose. It also catalyzes the conversion of lactose into allolactose. Now, as we'll see in the example on the whiteboard that we'll go to very shortly, allolactose is extremely important because it binds to the repressor protein, which enables RNA polymerase to bind to the promoter region to start the transcription of the structural genes. We then have lactose permease, which is coded for by the gene called LAC-Y, and lactose permease is a membrane protein that enables the uptake of lactose into the bacterial cell. Finally, we have transacetylase, which is coded for by the gene called LAC-A, and transacetylase is an enzyme that is thought to have the role of metabolism, of sugar metabolism and detoxification. So now let's go ahead and take a look at the example of the explanation of the lac operon on the whiteboard. So now that we've taken a look at the terms regulatory gene, structural gene, we've taken a look at the different genes within the lac operon, Let's now go ahead and take a look at how all of these terms come together in the lactose operon example. So the regulatory gene is constantly producing two binding sites. One binding site that binds to the operator, and one binding site to which it can bind to allolactose. Now, without allolactose present, the will be constantly bound to the operator region. And because the promoter and operator regions are very close to one another, they're adjacent, the promoter region is actually blocked. We can see it's blocked. And so RNA polymerase can't come and bind to the promoter region to start transcription of the structural genes. Now, as we know, beta-lactosidase will hydrolyze lactose into glucose and galactose, but it also catalyzes the reaction of lactose into allolactose. So that allolactose, when we have the allolactose present, it will attach to its binding site and that will change the binding site that used to bind to the operator, so it can no longer bind to the operator. This will then leave the promoter region uncovered for where RNA polymerase can come and attach to, and transcription of the structural genes can actually go ahead and take place. Now, it's quickly important to understand what is the promoter region. So the promoter region is simply where the RNA polymerase will bind to in order to start transcription of the DNA that we have of the structural genes. The operator region, as we saw here, works basically to switch on or off its subsequent structural genes. So now that we've taken a look at the regulation of transcription in bacterial cells, and we know from our classification and biodiversity video that we have three different domains, bacteria, archaea, and eukarya, it only makes sense that now we take a look at the regulation of transcription within eukaryotic cells. So, in eukaryotic cells, there are transcription factors. Now, most transcription factors are proteins, and what they do is that they attach to a section of DNA, and more transcription factors attach to the initial transcription factor, forming a transcription factor complex, to which RNA polymerase can then attach to and begin transcription of the subsequent genes. Now, when RNA attaches to that transcription factor complex, a transcription initiation complex is formed. Now, in higher eukaryotic cells, the number of transcription factor molecules required is very specific. So any missing transcription factor will result in the RNA polymerase actually not being able to bind to begin transcription. And so we can see that also, on top of being able to enable transcription to occur, transcription factors also have the ability of preventing transcription to occur. Okay, and the reason why we need transcription factors is because RNA polymerase cannot actually bind by itself to the DNA to begin transcription. So now that we've looked at transcription factors, let's look at gibberellin and its role in stem elongation. So in plant cells, there are proteins called proteins, and these attach to transcription factors and prevent them from enabling the RNA polymerase to begin transcription. So what happens with gibberellin is that it will attach to a receptor within the plant cell called GID1, forming a gibberellin receptor complex. And the ubiquitins will mark that complex for destruction by an enzyme called proteasome. So now without the delaproteins, the transcription factors will be able to do their job, transcription of the genes will be able to take place, and the enzymes and proteins will be able to be synthesized and which will then enable stem elongation. So that's it for this information-packed video. If you stayed around to the very end, congratulations, I hope you learned something. Otherwise, thanks very much for watching, and I hope you have a great day. Bye-bye.